Very good afternoon to everyone. Thank you for joining us today in our February month Research Edge series. I hope you all doing well and continue to be safe wherever you are. So today, once again, we have another interesting session that we're going to listen to two different kind of presentation. The first presentation, the Global Insect Apocalypse, developing a national insect monitoring program in the Bahamas. This is going to be presented by Dr. Paul De Luca, who is the assistant professor in biology at the School of Chemistry, Environment, and Life Sciences at the university. His presentation will describe a proposal to develop a national plan for monitoring insect biodiversity in the Bahamas. Following his presentation, we have another interesting international diverse team of presenters who's going to be speaking on mtDNA of ancient indigenous Bahamian islanders. So this presentation will be presented by Vanessa Forbes Pateman, Dr. Aram Yardumian, Dr. Miguel Villar, Dr. Tania M. Sims from UB, Dr. William Keegan, and Dr. Michelle Pateman. So we have a quite an international diverse team that is going to be presenting today. So without further ado, I am going to invite Dr. De Luca. Dr. De Luca, I see you're already online. So I'm going to allow you to share your screen now. Let me remove my screen. So you are free to share your screen and start the presentation. So try to limit your presentation about 20 minutes or so that we have enough time for discussion. Over to you, Dr. De Luca.
Thank you. I hope people can hear me.
Yes.
I appreciate having the opportunity to give this talk today. This talk grew out of an invite from the UB Environmental Pride Club last year to give a talk about the insect apocalypse, which was canceled by COVID. And I also developed this talk to give at the BNHC conference here, which was canceled due to COVID. So I do appreciate the opportunity to give this talk here today. And so I am just going to start my slideshow right there. I hope my video isn't in the way. I could turn off my video if it's a problem, but if not, I'll keep it on as I go. And so again, thank you, Dr. Nair, and thank you everyone for joining today. I do appreciate the opportunity to talk about some ideas I've been mulling in my head for the last little while about developing some insect monitoring programs here in the Bahamas. And when I go through this talk, I would like you to keep this term in the back of your head, the Anthropocene or Anthropocene. It is a proposed new geologic epoch that encompasses the commencement of significant human impact on the Earth. And so it is a new geologic epoch that has been thrown around, and it really has, they haven't figured out exactly when they may want it to begin, but it encompasses the time where we think humans have had a serious impact on the world. Anytime from the development of agriculture to maybe when the first atomic bomb was detonated in 1945. Because my talk today is about biodiversity loss. And again, a lot of the driving or proposed driving factors have got to do with human effects. And there have been a lot of reports, in recent years of big declines in biodiversity. A big paper came out in Science a couple years ago talking about massive declines in North American bird populations, surveys of hundreds of species, estimated 3 billion fewer birds in the last 50 years, and implicating certain things like habitat loss, increased pesticide use. And, not just birds. We have had similar reports for mammals. A big paper in PNAS just came out, similar big paper in Nature about big declines in shark and ray populations. And so we are experiencing, these massive drops in some very large groups of animals. And when we think about insects, though, we generally have not put insects in that category. You know, this image here is something called a speciescape, and the relative size of the organisms in this image refers to basically their diversity, the number of species in that group. And as you can see in the top left, as indicated by the beetle, you know, insects by far outnumber most other organisms combined in terms of the number of species. You know, you see the little elephant down there representing mammals of only about 6,000 species, whereas, you know, we have easily 1 million described, maybe 10 or 20 million undescribed species of insects. And so when we think about biodiversity loss, generally insects have not been included in these discussions. And certainly when you see things like this, I'm just going to play this video. I hope it comes through okay. You know, the recent locust plague that have affected Africa and the Western Middle East in 2020 and stretching into 2021. This is, I believe, just the church of Herbarium, one of the plague locusts that does affect Africa and Asia. Huge swarms of this have devastated farmlands and agricultural areas in Africa and Western Asia, and it's continuing in 2021. And so when you see literally billions and billions of insects like this, you might not be thinking that insects are in trouble. And so, you know, in this context, I'd like to say, well, we do have some data that I'd like to share with you that we are reporting some big declines in insect populations. And some of you out there might be like, well, declining insects ain't such a bad thing. I could do with a few less mosquitoes in my life. But of course, insects do serve some very important roles for humans and human society. You know, we should be concerned if insects are on the decline. For example, they provide very important ecosystem services. It's estimated up to 70% of the 1,400 species of agricultural crops insects provide pollination services for. And so they provide a very important service for us, keeping us fed. Many insects are recyclers, from these beetles breaking down dead animal carcasses to termites that recycle dead wood and forest matter. You know, without some of these nutrient recyclers, we literally would be neck deep in dead organisms or, you know, poop. And also biocontrol. We have employed dozens and dozens of insect species in our efforts to control agricultural pests. All right. So very important roles that insects play for us. Also, insects provide a lot of products that we like. Many of us like honey and pollen that we include in our diet, from the silkworm moth that makes silk that many of us may use, to some dyes that also insects produce. And the list goes on. I can teach an entire course really on the good that insects do for humans and human society. And then also the advances in science we have derived from insects, right? Our modern understanding of genetics and inheritance comes from the pioneering work in the early 1900s by Dubjansky with respect to genetics. And so there's really a lot of good here that insects do. So I'm going to put the argument out there that we should be concerned if insects are on the decline. And so I'm going to show you just a couple papers, certainly not exhaustive, of some recent studies that are reporting these long-term declines in insect abundance. The first is about a 30-year study from Southern England showing, up to a 75% decline in insect biomass. And so on this graph, you see the years on the X and biomass on the Y. And what they've done is they basically set up these big vacuum cleaners about 12 meters up in the sky that would suck anything that flies over. And so they mainly were targeting flying insects. And as you can see, they experienced quite a big decline over about 30 years. And again, the response variable here is biomass, right? So they just basically weighed everything, and that was their measure of abundance. A similar study from Germany over a similar time span also recorded up to almost a 77% decline in flying insects. Now, they didn't use vacuum cleaners here. They used what are called Malaise traps. And so this kind of funky looking tent here is a Malaise trap, where the idea is an insect will hit the trap, it will instinctively crawl up on the inside of the fabric and then get caught in these little catchment containers. And then you can empty it and look at what's inside. And so again, this graph on the right shows a drop in insect biomass. So in terms of abundance, right? Something a little bit closer to home in Puerto Rico on this side of the Atlantic. A similar study was published not too long ago in PNAS. And here again, they showed quite a large drop over about a 25, 30-year sampling period. And so they sampled in the mid-70s, they sampled again around 2011, 2012. And again, they found some big declines, whether it was from sweep netting, so walking around with a butterfly net and just catching things, or sticky traps, basically fly paper they would hang up, things would get stuck to it, and then they could measure and identify what was there. And so on this graph here, you could see again, a very large drop in, again, the biomass, the abundance of insects in these traps. Now, the interesting thing about this study is they didn't just focus on insects, they also focused on predators of insects, lizards, frogs, birds. And this middle graph shows the result for lizards. They found a very similar decline in lizard abundance, and the same thing for frogs and birds. And so when you get these parallel declines in all these different, trophic guilds, they basically were quite concerned and found, we're finding basically a collapse of an ecosystem here that they tie to the loss of insects. And their implicator here was temperature. On this last figure on the right, you could see the rise in mean temperature over this sampling period for Puerto Rico. And so their argument was, global warming was basically the driving factor in this trophic collapse, okay? And so those were just, a couple papers out of several that have come out in the last few years tracking insect declines. And a recent review paper that basically gathered data from about 73 reports that found these declines in insects. They tried to find, what is driving the decline? What are some of the potential causal factors? And in this review paper, they basically label habitat loss. And so this box here on the right that I've highlighted, really about 4 of the 6 or 7 major implicators have to do with habitat loss. And so again, thinking about the Anthropocene and some of the factors, human cause factors that may be driving this decline in global insect abundance. And of course, I'm just talking about good old normal climate change. And on top of that, we now have the new influence of what some scientists are calling extreme events, okay? And so, human actions through climate change is causing, in some instances, increases in the frequency of what we're calling extreme events, all right? Extreme weather events. Now, these are basically unpredictable, high magnitude events that are, over and above what we're expecting from, what I'm going to say is normal climate change. The severity of droughts, the severity of fires, the severity of hurricanes is increasing. And some of this is also very unpredictable and hard to control. And some of the images here just showing you on the left, the terrible wildfires that hit Australia a year ago, the big tsunami on the right from I think it was the 2011 Fukushima nuclear disaster, the earthquake that affected that and caused a large tsunami in Japan. And so these are the kind of extreme events that I'm talking about. Now, the really cool thing is, just last year, an opinion piece in an insect journal, some researchers came out and they tried to develop a framework to incorporate these lethal, unpredictable, high magnitude events. And so they came up with this acronym, a shock, a sudden high magnitude opportunity for a catastrophic kick. I love that term. And so they developed a framework. And what I really liked about this paper is, trying to incorporate at the bottom here, they've got some of these shocks, whether it's a natural shock, a tsunami, a volcano, to a human-mediated shock, a nuclear disaster, a dam collapse, or something like that. And so they then identified several ecological or life history variables that might be affected by these kinds of shocks. And they developed this paper in the framework of pollinator conservation, right? Looking at bees or butterflies. But you could really apply this framework to any other group you might be interested in, right? And so, this brings me to the Bahamas. You might be then wondering, what about global collapse or global insect collapse for the Bahamas? And I do give you here or show you a picture of Grand Bahama right after Hurricane Dorian. This is Lucayan National Park right here on East Grand Bahama. Some of you may know Lucayan or National Park or visited it. And so here is, the parking lot right here, if you could see my pointer, the boardwalk down to the beach. The beach was utterly destroyed. But what is more striking and what blew me away when Mr. Ellsworth Weir from Rand Nature Center sent me this photo is just the devastation, right? From the southern shore to the north. The absolute devastation of the pineland, the salt inundation from Dorian, basically burning and destroying literally hundreds and hundreds of acres of forest land. And so, the truth, or the short answer really is we just do not have good historical baseline data on insect populations for the Bahamas. There have been a couple studies, mainly focused in the exumas that I was able to find, where they did a biodiversity assessment a few years ago, which is great, but that's it. There was no follow-up. It was what we call a single time point assessment. The researchers published their data and they never came back, right? And so we really do not know. We have nothing to compare even, data we might get now, which doesn't mean we shouldn't collect the data now, but we don't have some, baseline data to compare it to. And really the effect of these extreme events, and of course, what is the extreme event for the Bahamas? Well, of course, they're hurricanes. And Dorian sort of highlighted the severity of storms we are experiencing in recent years. As oceans are getting warmer, it's providing the fuel to drive these really, really violent hurricanes. And, we have some data. The Bahamas National Trust has done bird surveys after Dorian on Grand Bahama and Abaco, but we really don't have any data on how our insect populations might be affected. Now, I thought about that and thought, well, could there be other shocks, other places that have experienced a shock that do have data? And I was surprised to find, yes, there was a really, I'm going to say, a cool study that came out just a couple years ago from Oklahoma, where these researchers were sampling invertebrate communities in a grassland habitat. And then unexpectedly, there was a massive spring flood, as you can see from this picture, that basically leveled the area. And so they were in a perfect position to actually sample before and after this big unexpected shock. And they found some really neat things that potentially we could use to make some predictions for the Bahamas. Basically, they found massive declines in abundance and richness. And so abundance, how much stuff, how much biomass was there, declined over 90%. And richness, basically how many different species were there, declined by about 60%. They found some interesting changes in the functional guilds, right? So above and below ground communities became similar. What that means is a loss in ecological specialization and ecological guilds in above and below ground invertebrate communities began to homogenize. They found scavengers and detritivores recovered quicker. Maybe not all that surprising. After a big flood, there's probably a lot of dead vegetation, dead organisms. And so other organisms that specialize on scavenging probably would do very well after a big event like this. And they also found, for example, invasive species like fire ants did really well. And that has implications for us too. We do have fire ants, we do have invasives. And one thing that makes invasive species invasive is they have a lack of predators. They are sort of in a new spot. They're able to really take root and outcompete other things because being new to the area, they may not have natural predators and things like that. A couple other things they found too, ecosystem engineers, things like ants, millipedes, isopods, things that turn over nutrients, nutrient recyclers, are very, very important in mediating how quickly an area can recover from a shock. And also, what is very important for the Bahamas as sort of an isolated island nation is the recovery after a shock is really dependent on where things are coming from. So dispersal from nearby sources of organisms that might migrate into the area. And so that also has implications for the Bahamas, right? And so thinking about this example from Oklahoma got me thinking about the Bahamas and some predictions that we might be able to make for here, right? So we might think that because of hurricanes and salt inundation like you saw from Grand Bahama, below ground communities may take longer to recover, even above ground communities from all the salt damage in the pineland or the coppice forest or things like that. We may find that our scavenging or detritivore invertebrate species may do well after a shock like a hurricane. And even we should focus on identifying invasive species. I talked about fire ants. We do have fire ants here in the Bahamas. We do have other invasives that maybe we could focus on as sort of target species after a shock to see how well they recover. And also, you know, being an island nation, our recovery time after a shock could be dependent on, you know, the recruitment we get from other areas. The biodiversity of the Bahamas, both plants and animals, is historically come from Cuba and Hispaniola, to a lesser extent, Florida. And so, you know, how well our insect or invertebrate communities recover after a Dorian or a similar shock, you know, may determine on what the levels of recruitment are from these other areas. Are humans moving things around? Are things rafting on debris? And also, of course, the recovery may be dependent on how other things recover, right? Insects are really, really obviously dependent on the plants. So how quickly plants may recover after a hurricane event and begin to flower again may have a lot to do with how quickly, you know, insect populations may recover. And so it is in this framework of shocks and insect declines that we've been reading about elsewhere that got me thinking about this plan, this idea of developing an insect monitoring program. And I've identified two phases or components of this project. The first is to revive and utilize our insect collections in the Bahamas. Of course, you know, biological collections, what are they? They're more than just a box of insects or plants that are in some dusty, dark drawer in some museum. These are repositories of biodiversity. They provide a link to the past, baseline data of diversity and abundance for a given geographic area. These are used for research, for teaching, and just for general posterity to know what was there at a certain time or certain point in history. And we are lucky in the Bahamas that we have several large insect collections. The national collection at the Botanical Gardens in New Providence, the collection at the University of the Bahamas that I will talk about in a sec. Jarris Research Center on San Salvador has a large collection. The Leon Levy Native Plant Preserve on Eleuthera has a small collection that is growing. And Rand Nature Center has a very nice collection that I recently was able to examine And I'll talk a little bit about that one as well. And really, the broader goal of this project is to establish the University of the Bahamas as a national center for entomological expertise and research and knowledge. I will say, though, that some of our collections in the Bahamas, they need some TLC. These are some pictures of the state of some of our collections here in the Bahamas. These metal drawers on the left are the insect collection, and these large greenish drawers surrounding it are actually the National Herbarium plant collection. And, you know, we do have challenges in the Bahamas. Our weather, especially in the summer, is a challenge. Keeping things temperature and humidity controlled is a challenge in a place like this. This drawer here on the right, you could see a lot of damage. All the little bits and brown bits and stuff is a result of pests that get in, other arthropods that may get into the collection and feed off the pinned insects can be a problem. And, you know, what's also very important is properly trained personnel. The truth is we have a challenge here in the Bahamas of properly trained personnel in curation and collections management that can keep temperature and humidity and pests and things like that at bay. And so this brings me to the insect collection at UB. We do have an insect collection, and I am in the process of updating this collection. I was awarded an annual fund in 2018. Unfortunately, this work was put on hold last year when COVID hit, and I hope to resume it in the upcoming months as maybe restrictions ease and there's a little bit more freedom of movement. But this is the UB collection. It's in these old wooden drawers that have got some termite damage. These drawers don't seal as well. And so with generous support from the annual fund, I was able to buy more properly sealed and good drawers. So I am in the process of transferring the collection over. And I do want to say that, you know, my entomology class is going to take a lead role in helping as they take this course. One thing I would like to do is to train them in things like collections management. The second phase, or the second part of this project, is to keep the collections alive and current through periodic collecting in the Bahamas. And I have initiated some collaborations with the Bahamas National Trust here. All right. In 2013, I established the small collection at the Levee Preserve, and I'm involved in periodic collections there. And in 2018, I taught for a semester at UB North prior to Dorian, and I initiated a collaboration with the Rand Nature Center to also update and curate their insect collection, but also to conduct periodic collecting on Grand Bahama and Abaco. Unfortunately, Dorian put a little halt to that. COVID put a little halt to that, but we hope to resume these projects in the upcoming months. But I would also like to say is this also must be an international collaboration. There are individuals that have been doing insect collecting and research in the Bahamas far longer than me. And so this truly needs to be both a domestic and international effort. Dr. Nancy Elliott, that some of you may know, she established the national collection at the Botanical Gardens in Nassau exactly 20 years ago. She has done so much work with the collection at the Jarris Research Center. And I'd also like to highlight Dr. Debbie Lott from the Florida Museum of Natural History. She works on Lepidoptera with colleagues there, butterflies and moths. They've been doing work in the Bahamas for years, and they've amassed an absolutely amazing database of over 1,000 Lepidoptera species distributed across the Bahamas. And so my goal in you know, even establishing UB as a central resource for the Bahamas, but that will also include collaborations with our international partners that have done so much entomology research here in the Bahamas. And what I'm really excited about is we are at such a cool spot. I know I'm starting to get, animated here, but we are really at a good place with 5 really good collections. We are at a cool time now where we could start looking at these collections, evaluating inter-island diversity of these collections, and actually posing hypotheses and making predictions about insect diversity. This is just, a couple simple pie charts that myself and my collaborator, Dr. Kristen Brochu, compiled a couple years ago, where we examined the collection on Eleuthera, San Salvador, and Grand Bahama, and we just started putting together a database of the different groups of insects that were in these collections. And this allowed us to get some preliminary data on diversity in families and species just on these 3 islands, and I want to increase that to look at the UB collection, to look at the national collection, but then also to look at other islands that maybe have not been sampled as much as the ones here. So this is a really exciting time for doing entomology research in the Bahamas. Very exciting. And I will say that, again, a big part of this will include student involvement, whether it's my entomology students, whether it's Bio 480 students that I want to include in this work. I've just got a couple pictures here of my entomology class. We went in 2018 and 2019 to San Salvador to do collecting, and it was fantastic. I can't tell you how excited I was to see the enthusiasm of my students, whether it was collecting, whether it was the pinning and the preparation of specimens. They were really, really excited. They had a wonderful time. And what I want to instill in them are these skills in field collecting, collections management, species identification, because this isn't just skills for a course. These are skills these students can take when they graduate from UB and go off to bigger or better things, skills that they could use in conservation, environmental science, ecotourism fields that are growing in the Bahamas, graduate work they may go off and do. And so training them in these important skills is going to really give them, I think, a good advantage in possible upcoming careers like ecotourism, which is getting more and more popular in the Bahamas. And I would like to give a shout out to the Levee Preserve here. For several years, their internship program, many UB students have been part of their internship program. They have taken a lead role here. They focus on botany mostly, but they also do birds and reptiles. And they have trained many students in these exact skills. And so again, collaborations and partnerships with other domestic groups are going to be very important in training these students in all these diverse fields.
Okay, Luca, you may want to conclude. We're
Yes.
off the time.
We are done. I want to thank you. Thank you, Dr. Nair, for bringing me. I am done. I just want to acknowledge these folks. And afterwards, I'd be happy to take any questions. Thank you very much.
Thank you so much.
I will stop the share.
There we go. Interesting presentation. I think we can straight go to the questions since we are in the flow of the presentation. There's 2 questions over here. Let me start with the first question here. Are you familiar with the citizen scientist concept? Would a similar project across the Bahamas address the need for long-term data collection, not only insects, but also the supporting systems such as meteorology?
Absolutely. Citizen science is a great way to be monitoring our natural populations. It's used a lot to monitor bee populations for pollinator assessments in Canada and the States. And incorporating a citizen science component to this, yeah, absolutely would be very, very useful.
Another question here. Insects are not exactly popular, and most people's first instinct is to eliminate them. How can you emphasize the importance of their roles in the greater ecosystem and impact on climate change?
Thank you. That is the first question I ask my entomology students before I do anything, is I get them to list some words that come to their mind when I say insects. And you usually get some negative words, some I can't repeat. But by the end of the class, it's amazing how it's changed. And what I do is I make sure to stress the important roles that insects play in society, like I talked about early on. And I think when people grasp something like 1 in every 3 bites of food is provided to you by a pollinating insect. Think how much blander your diet would be without the food a pollinating insect provides. And so when they start to think about insects in that way, they start to, I think, garner a new appreciation. They still don't like cockroaches and mosquitoes, and that's okay. But I think they do appreciate some of the broader roles that insects play.
Yeah.
Thank you. That was a great question.
Another one, it was mentioned insects go underground during a natural disaster like a hurricane. Do they burrow in the soil or into caves, perhaps?
Wow. What a wonderful question. I have no idea. Where the insects go during a hurricane, where do all the other animals go? The birds, the snakes, the mammals, they find refuge somewhere. Insects probably go deep in the coppice. They may go underground in caves. That would be a great study to do to find out where they go. That's a great question. I am not sure where they go, but they go somewhere. They go somewhere.
Okay, we have one more last question. Due to the decline of insect, has there been any changes in insect features or adaptation to their environment?
Ooh, what a great question. I am not aware of any. There may be some studies, but in the short time frames we've been talking about, we may not see any evolutionary or phenotypic change to insects. There may be some studies out there that address that over a longer time scale, but right now I'm not aware of the short time frames of decades we're talking about causing any particular evolutionary changes. But there may be papers out there that might address that. Thank you.
Okay, there's one more just came in. Have you traveled to island of Andros for data collection, since it is the largest island in the Bahamas and can present itself to give a good data and many unknown insects?
Yes, thank you. I've been to Andros many times. It's probably arguably my favorite island. I've never done collecting there. I would like to incorporate Andros because it is the biggest island and it deserves to be explored in a proper way. So plans are in the works. Plans are in the works. Thank you.
Thank you. Thank you so much, Dr. De Luca, for an excellent presentation. And thank you for all the questions. If you have any more questions, feel free to email Dr. De Luca, or you can even email me, the research center, and we will send the question to Dr. De Luca. All right. So we move on to our second presentation today on the mtDNA of ancient indigenous Bahamian islanders. So today's session, we have 6 presenters, and I'm great to see that all the 6 presenters are here today. So this session is going to be led by Ms. Vanessa Forbes Pateman. She's a PhD scholar from the Simon Fraser University in Canada. So I'm going to allow Ms. Pateman to introduce your team and also share your screen and coordinate the presentation among your team. Over to you.
Good afternoon, and thank you for inviting us to this presentation. I'm actually not Ms. Pateman. I'm Dr. Pateman, Michael Pateman, and I will be the one to introduce the project and introduce our colleagues. So the lead author on our project is Vanessa Forbes Pateman, who's a PhD student at Simon Fraser University. We also have Aram Yardinium, Miguel Villar, Tanya Sims, who's at UB, Dr. Bill Keegan, and myself. And our focus is on, our study evaluates the genetic ancestry of Lucayans and their relationship with other pre-Columbian and contemporary Caribbean populations. So I'm going to give a brief introduction to the Lucayans, and I'll be followed by Vanessa, who will discuss our samples and our methodology. And then Tanya will present our results and conclusions, and then the rest of the team will be available for any questions that maybe anyone has. Okay, so who are the Lucayans? Or who were the Lucayans? The Lucayans, or the Lucukari as they refer to themselves, were the people who Columbus first encountered when the Europeans stumbled into the New World. The Lucayans were also the first victims of large-scale genocide of indigenous people in the New World by the Europeans, with most historians suggesting an extinction by about 1520. However, new research may change our thinking on those dates, but that's not the focus of this presentation. So where did they come from? There are two major migration waves into the Caribbean. The first is categorized by a migration from about 6,000 years ago and is associated with archaic people, and they were from Central or South America. However, this period is still not as well known as some of the other periods because it's not really well studied or adequately dated though. Most of the sites are in Cuba, Hispaniola, and Puerto Rico, with evidence of some sites in the Lesser Antilles. There was a second migration wave, which started about 2,500 years ago, and is distinguished by the introduction of pottery and agriculture by an Arawak-speaking people from South America. They reached the Antilles about sometime between 1,700 and 2,000 years ago. So where did they come from? Continued. The Bahama Archipelago, and we'll call it the archipelago because it includes the Turks and Caicos Islands, bears a unique distinction amongst the Caribbean archipelagos of being the last to be populated by the pre-Columbian people. But interestingly enough, it's the first to be encountered by Europeans. The earliest migration into the Bahama Archipelago occurred about 700 AD, with the earliest known site on Grand Turk in the Turks and Caicos Island. These people came from Hispaniola. This early site is shown to be the location of seasonal exploitation for resources. However, shortly after this, we had a migration of permanent habitation sites, also from Hispaniola, about 700 to 900 AD. Additionally, based on evidence in the Central Bahamas and the Lucayan names of the islands, it is also proposed that a separate migration took place from Cuba around 700 to 900 AD. How do we know that Lucayans were here? Well, we find their sites throughout the archipelago. These sites include some of the examples we have up on the screen, which includes caves that has burials or some of the rock art. For example, a famous rock art site is Hartford Cave in Rumkey. Also, we find habitation sites and resource exploitation sites with pottery, tools, and other material.
So I'm going to take over now, and I'm going to talk to you about the sample collections. The human remains included in this study were curated at either the Yale Peabody Museum of Natural History or at the Antiquities, Monuments, and Museums Corporation. All 31 of the skeletal remains of the ancient individuals in this study were secured from the museum collections in the Bahamas. The work was performed in dedicated ancient DNA laboratory clean room facilities at the Harvard Medical School, the University College of Dublin, and the University of Vienna. The remains were disinterred from wet caves, dry caves, or sand dunes, including the following. If you look up on the screen, going from top to bottom, the burial locations and samples are listed to the left of the screen. Abaco, Hopetown had one female found in a sand dune. Bill Johnson's cave had one male. Randy's cave had one female, and Imperial Lighthouse Cave had one male, for a total of 4 sites and 4 samples, 3 of which were found in dry caves. For Eleuthera, Preacher's Cave, there were 4 females and 1 male. Valentine's Cave included 1 female and 1 male. Garden Cave included 3 females, and the Wim's Bite Caves included 1 male, for a total of 4 sites and 12 samples. For Andros, Sanctuary Blue Hole had 1 female and 6 males, and Stargate Blue Hole had 1 male, for a total of 2 sites and 8 samples, 7 of which were found in Sanctuary. Ragged Island on Flamingo Key, there was 1 site and 1 sample was found, and that was male, and that's a dry cave. On Long Island, we had Clarencetown Burial Cave, which included 1 female, Rolling Heads, which included 2 females and 1 male, and they were found in a sand dune. So that's a total of 2 sites, 4 samples, 3 of which were found in a sand dune and 1 in a dry cave. For Crooked Island, we had Gordon Hill Caves, which had 1 female, Burial Cave, which had 1 male, for a total of 2 sites and 2 samples, and they were found in dry caves. So here on the screen, you'll see the Rolling Head site, which is LN 101 in Long Island. And to the right of that, you'll see Sanctuary Blue Hole, and that's in Andros. For our methodology, in this research study, we used a lot of sciencey words and phrases. So you'll hear terms like radiocarbon dating, DNA, whole genome sequencing, phylogenetic trees, haplogroup assignments, MDS studies, and networks. I'm going to try to explain why these techniques are used and what they actually mean. Before I begin, I must note that the mtDNA and the Y chromosome haplogroups presented in this work were submitted as a part of 3 previous projects that mapped the genome of ancient pre-Columbian people in the Caribbean. And in our study, we reanalyzed that data to formulate our own hypothesis and conclusions about how the islands of the Bahamas were populated during pre-Columbian times. Up on the screen now, you'll see the samples that I actually talked about previously, and they were radiocarbon dated. Radiocarbon dates of the bone fragments were generated using accelerator mass spectrometry, pardon me, AMS, at the Pennsylvania State University Radiocarbon Laboratory and several other labs. The samples were prepared by extracting and purifying bone collagen, followed by ultrafiltration. If the collagen yield was low, a modified process was used. All the dates for the research were reported as calibrated years before present. So for unprecedented inherited marker system, i.e. the mitochondrial DNA, if you look up at the screen, there's a, you can see the mitochondria up in the top of the screen. If you look at the cursor, I'm pointing at that, which is found outside the nucleus and inherited from our mother, and the Y chromosome, which males inherit from their father, are useful for tracing the patterns of female and male migration into a particular region, and in this case, it's into the Bahamas. To do so, mutations or permanent changes in the DNA sequence that are polymorphic differ, and different forms exist as typed. The information obtained by typing a series of markers on the mitochondrial Y chromosome is used to determine your specific haplogroup. I know you're asking yourself, what is a haplogroup and how does it pertain to your family history? Well, a haplogroup is a group of individuals that share a common set of DNA mutations. Haplogroups, which are population specific, are named alphabetically in order of their discovery. For the Y chromosome, there are 20 different types of haplogroups, A to T. If we look at the haplogroup Q up on the screen, the black arrow points to Q. It's denoted, it is denoted in purple. For example, we see that it is found at high frequencies in North and South America and at low frequencies in Asia. This haplogroup has often been found to originate in Central Asia and then spread to Siberia and then to the Americas. This shows that knowing which haplogroup you belong to can tell you where your ancestors came from thousands of years ago. Mitochondrial haplogroups. Likewise, for the mitochondrial DNA, there are 26 different haplogroups, A to Z. Again, if we focus on the North American haplogroups, which are denoted by A, B, C, and D, the arrows are on the screen pointing to them. These groups are also found at high frequencies in North and South America. Interestingly, because the mitochondria undergoes very little recombination, you will share the same material, the same maternal haplogroup with any relative that you share a direct maternal line with. The same holds true for the Y chromosome. Even though it does undergo recombination with the X chromosome, but this only happens at the end. Thus, the majority is unchanged. Thus, a male would share a paternal haplogroup assignment with any male relative that he shares a direct paternal line with. As a result, we can use the mtDNA and the Y haplogroups to tell us where the Lucayans came from before they migrated to the Bahamas and where they settled once they colonized the archipelago. To assign haplogroups to the mtDNA and Y chromosome, we had to test for different mutations. Shown here is what we call a phylogenetic tree, a hierarchical representation of DNA mutations. If a person lacks the mutation, they are referred to as ancestral. But if they have the mutation, they are designated as derived. If we go back to the Y haplogroup Q, remember we showed that to you on the screen in purple, for example, we would first test for the presence of or absence of the defining marker M242, which is here in red. If the individual had the mutation, they are considered derived. We would then move down the tree to test M378. If ancestral, then we would test MEH2. If derived from this marker, we would test one of the downstream markers, either M450, M25, or M120. If ancestral, we would move to the CIDR branch and test for M12. If derived here, the sample is considered to belong to Q1A1A1. So further, let me explain to you what an MDS is. An MDS is a multi-dimensional scaling graph in a two-dimensional plot that is generated using genetic distances between the pairs of populations. The aim of the MDS analysis is to represent high dimensional data in a low dimensional space with the preservation of the similarities between data points. In this study, we use the MDS to graphically show representations among ancient and contemporary populations from the Lesser and Greater Antilles, as well as South America, that may have migrated to the Bahamas. It should be noted that this graph has to be performed at a low resolution to be able to include pertinent populations. The program used to generate pairwise FTS distances was Harlequin version 3.5.2, and the plot itself was constructed using PASW statistics version 25, aka SPSS, as most graduate students know it. And I just want to point out that for the network, also, it was used to examine the genetic diversity presented in the ancient Bahamian samples and to determine if gene flow into the islands from the other greater or lesser Antillean groups occurred. The network analysis was performed using the network version 10 program for the two major haplogroups found in the archipelago. Any established patterns have variously been attributed to the anthropogenic introduction associated with historical cultural connections, population expansions caused by a replacement of intervening populations, or a population bottleneck causing a loss of connecting haplotypes from the intervening populations. And I'm going to hand this off to Tanya, who's going to explain what I talked about.
Turn on your mic, Tanya.
Sorry. So good afternoon. How's everybody doing? Now we're going to get to kind of the nitty gritty. What is this? What does all this jargon actually mean? What do we find? Well, if we look at the Y chromosome haplogroups, you'll see that we have pretty much one color. right? And we have a little bit of green. So what that means is the groups that came up into the Bahamas had to, they basically had haplogroup Q that Vanessa talked about. So they had that defined a mutation, but then they developed a few other mutations. And basically what we see is a haplogroup Q1B1A, which is defined by M902. And in Andros, we actually see a sister clade to that Q1B1A, which is called Z780. So the Y chromosome is pretty much homogeneous in the Bahamas, right? Then we go to the mitochondrial DNA. And now you're gonna see that the colors kind of shift. We have more diversity present here. So notice that Abaco and Crooked Island are pretty much homogeneous in terms of the mitochondrial DNA that they have, right? So we see Abaco has just orange, which is B2, and Crooked Island has just gray, which is C1b. But the other groups, Andros, Eleuthera, and Ragged Island, actually are gonna have those two haplogroups in addition to two other groups, A2 in Andros and C1d in Andros and Long Island / Ragged Island. So the mitochondrial DNA is definitely a lot more diverse. So then what we did was we looked at the dates, the radiocarbon dates. And what we found is that the skeletal remains range in date from about 500 before present to 1200 before present. If we then separate those samples into mitochondrial haplogroups, you'll notice that haplogroup B2 and haplogroup C1d tend to be the oldest, of those two, B2 being definitely the oldest, followed by C1d. C1b is kind of around the age of C1d, a little bit younger, but then A2 is definitely the youngest. So we do see a difference in the dates of the samples that were collected based on haplogroup. And I'll come back to this in a little bit. Then the next thing that we did was we performed what's called an MDS, as Vanessa explained. In this MDS, basically, the closer two populations are together, the more genetically related they are. So if you look at this graph, you notice that we kind of have one cluster on the left, and we have one big cluster on the right. The cluster on the left is going to be have Venezuela, Abaco, and indigenous group, an Arawak speaking group from Peru, Bolivia type region, okay? So Abaco is showing genetic affinities to the ancient Venezuelans, which is what we expect because we know that was kind of the route that we've traditionally learned in high school, that the Arawaks came up from South America, right? If we look on the right, you'll see that there's now one big cluster. Within this cluster, there's definitely a subdivision. Can you go forward, Michael? Sorry. So there's definitely a subdivision. One more. So you'll see one more. So you'll see kind of two different clusters here. The one to the top, so what we're focusing on is this blue kind of pentagon, sorry. The blue pentagons are gonna be our Bahamian samples. So the samples from Andros actually are gonna be most genetically related to ancient Puerto Ricans, but they do display some genetic affinities with modern and ancient groups from Puerto Rico, Guadeloupe, and St. Lucia. In the bottom cluster, we see Eleuthera and Long Island, Ragged Island clustering together with a very indigenous group from Brazil. So they haven't had any, even though it's a modern group, they really haven't had any genetic admixture. So they're kind of representative of the indigenous people in that region. And we see some affinities to Cuba and ancient Cubans and ancient Guadeloupe. So this is kind of showing genetic affinities as well to the Lesser and Greater Antilles, as well as to Brazil. And you also see affinities to this St. Vincent modern. Now, why this is particularly interesting is because the group from St. Vincent is what we call the Guafarina. The Guafarina is an African group that is mixed with indigenous Native Americans. So that's why we kind of see that clustering there. Crooked Island is kind of on its own. This is because it is solely C1b, I think. Can't remember, C1b or C1d, but it's only 2 samples as well. So that's kind of why it's kind of separated from the other groups. Can you go to the next slide? So then what we did was we performed a network. So what the networks are is basically they're going to show genetic diversity within a single haplogroup. So the MDS looked at all the haplogroups. We looked at the major haplogroups A, B, C, and D, but with the networks, we're kind of specific to one haplogroup. Also, the network was created at a very low resolution, so we can include pertinent populations to see genetic relationships. But with the network, we've done this at a much higher resolution. As a result, you'll notice that there aren't many reference populations included. For the networks, all the samples were ancient except for Puerto Rico. The reason why we kind of had to include Puerto Rico in this network is because this lineage here that I'm going to talk about in a minute is actually very rare in the Caribbean, but we see it tremendously in the Bahamas. Okay. So basically what this network is telling me is that there are 2 branches from a common root. So we have a B2 branch over here, which is Bahamian specific. It has Eleuthera and Abaco. And then we have this other cluster over here, which has, again, is a branch off of this common root. But notice that we have this bigger, larger cluster of 5 Bahamian samples. So these 5 samples have the same set of mutations. Some of the samples then developed a further mutation until you see this line kind of originating from here. So now we have Abaco and Eleuthera developing now a further mutation from the majority of the Bahamians that have this haplogroup, as well as a sample from St. Lucia. So this is showing that there's pretty much both haplogroups present in the Bahamas, both B2 and B2E. And there's a lot of diversity because we do see, you know, lots of mutations kind of originating from a common source, okay? The other interesting thing about this cluster in particular is the program will allow you to do a time estimate. So we did age estimates based on radiocarbon dating, right? But the program will also sometimes, if the data enables you to, will give you an age estimate. And for this haplogroup, we actually got an age estimate of 2,070 years, plus or minus 1,035. So this is about 2,000 years old. Yeah, the error is kind of big, but we have such few samples that we kind of have to go with it. But it just kind of confirms that this is probably the oldest haplogroup in the Bahamas. Next. So then we did the same thing for the next most common haplogroup, which was C1. So we had both C1b and C1d, as you saw in the map, right? So we did a network based on this haplogroup. And again, we have a common root, but now we have two branches as well. So we have a C1b, we have a C1d, and then we have a C1d1, okay? So the C1b, the Bahamians are kind of all dispersed in between Curacao, Puerto Rico, and Hispaniola. Again, that's showing some genetic affinity to these greater Antillean populations. And what's really interesting is this branch here. The Bahamians from Eleuthera and Crooked Island actually have the same haplogroup, the same set of mutations as those from Hispaniola. So that definitely shows even more genetic affinity to the Greater Antilles, particularly Hispaniola. Then 2 of the Bahamian samples actually developed a new mutation, and from that mutation, another sample developed an even further mutation. So basically, these networks are showing based on specific haplogroups that we do have genetic affinities with those from the Greater Antilles. Okay, next. So what does all this mean? So why am I telling you all this? Well, in terms of the mitochondrial DNA, if we look at, we know we have haplogroups B2, C1d, C1b, and A2. If we look at the radiocarbon dating, we see that haplogroup B2 is the oldest lineage, and this is going to be followed by C1d. Both B2 and C1d are present in most of the archipelago, all right? B2 probably came directly from up the Lesser Antilles from South America. And we actually have a sublineage called B2E that is pretty much unique in the Bahamas. But again, I say that with caution because we don't have every Caribbean sample, okay? We do see one other sample from St. Lucia that has it, but of the samples that we tested, B2E is pretty much unique in the Bahamas. C1d is a little bit younger than B2. It's also present kind of ubiquitously throughout the islands examined. And we see that this may actually be linked to Cuba or Hispaniola. Now, C1b is a little bit younger. And what we think happened is, again, this spread up the Lesser Antillean chain from South America. And this probably likely replaced some of the B2 that was originally there. And that's why we see it a little bit more in the south than in the northern islands. And then finally, we have A2. A2 is actually the youngest lineage that we find. And we see that this is actually connected to Cuba, Puerto Rico, or Hispaniola. And again, this is thought to have replaced some of the B2. Now, remember, A2 was only found in Andros. So we think it came from the Greater Antillean route into Andros more recently than the other haplogroups. Next. Now, unlike the mitochondrial DNA, the Y chromosome is actually really homogeneous. It's found in all the islands examined have haplogroup Q. All of the islands examined have this Q1B1A. So everybody has this. This all came up from probably South America. But Andros kind of has this Q1B1A2. So if we look at this tree here, everybody has this one, but Andros kind of has this one over here. So it's a sister lineage. And we suspect that that probably came from Cuba. Next. So in addition to looking at the mitochondria and Y chromosome, we also had some results on what we call relative matching. This is a pretty new technique where long strands of DNA are basically compared. And the more DNA that is shared in these long strands, they're referred to as genetic cousins. And what we found is that Bahamians actually have a cousin with Cuba and 12 cousins with the Dominican Republic, 2 of which also match Puerto Rico. So again, this is definitely showing us a greater Antillean connection in the Bahamas. Next. So in total, altogether, what does this mean? So this means that Bahamians, yes, we received migrations up the route that we've always learned in high school from Venezuela up the Lesser Antilles into the Bahamas, right? But we also seem to have received some genetic contributions from other Greater Antillean populations. So we have strong ties to both Hispaniola, Puerto Rico, and there seems to be some links to Cuba. There's definitely that B2E connection with South America. And remember, this is the oldest lineage, so this probably came up the chain first, and it was eventually replaced later on by C1d and C1b. And we definitely, I know you're like, wait a minute, we're so close to North America, we have to have gotten something from Florida. Well, we didn't find that. There's no, connection to Florida. Next. So altogether, this is kind of what we found, but we don't have we don't have every population in the Caribbean. So we can't make definitive conclusions because we don't have all the data. So this is telling us that we need to do some more sampling, right? We need to get some other Bahamian samples, maybe Grand Bahama, maybe in the south. We need some more from maybe like Mayaguana, Inagua. We need some Turks and Caicos. Jamaica, there's basically nothing, so we need some samples from Jamaica. Haiti, the samples were very limited, hence they were kind of combined with Dominican Republic. And then some of the lesser Antillean populations are still unavailable. So further analysis is still needed. Next. So for this study, I would like to thank, first of all, for all of you for coming and listening to us, for Dr. Vic for allowing us to kind of present today. Also, the authorization letters for the sample collections was signed by Michael Pateman, Dr. Michael Pateman, who's the director of the Turks and Caicos National Museum. The letter of permission for the export of samples from the Bahamas and for ancient DNA, isotopic and 14 carbon dating analysis by Dr. Keith Tinker at the National Museum of the Bahamas. Dr. Michael Pateman had a grant from National Geographic Society, and Dr. Aram Yardumain from summer research funds from Brian Anthon College. Next. So I open the floor now for questions. Thank you.
Thank you so much for really an interesting presentation and interesting research, and great to see this collaborative work that you have actually put together here today. So really fascinating. Let me see if there's a question here. There's one question here from Dr. Nicolette Bethel. First one of this, given the lack of evidence for a connection with Florida, was there not a burial site in Grand Bahama that might be able to provide material for testing?
So I can answer that. So yes, there was some burials found in Grand Bahama, but the challenge is with a lot of things found in the Bahamas before the Antiquities Corporation was in existence, where are these remains today? And I've spent a lot of time trying to figure out where they are. It seems like they may have been reburied. So we're looking at possibly going back to try and sample these along with another site that we know about in Grand Bahama. And we have other locations of where samples are. So we're looking at doing some further studies so we can expand our results.
Okay. More questions? Any more questions from the floor here? Mr. Wish, you want to ask some question? You want to directly ask, then I can unmute you and you're free to ask your question.
Dr. Vic, I sent you a message in the chat group, and the question was, would any of this study have been possible without access to the genetic resources? Thank you. Or that is, in this case, bodies.
No, we needed the burials to do the DNA analysis. And I know this is actually a hot topic in the Bahamas right now because the government just passed the
Yes.
the legislation. I can't remember what it's called, but this study was actually approved right before all of that was put in place. But I'm excited about the access benefit sharing. That's it. Legislation that's being put in place and future studies with that. And I see Nicolette had a second question about the underwater burials, how and why the Lucayans did it. That's the million-dollar question, and something I know I've been trying to figure out. Bill Keegan is on the call, and I don't know if he can answer that any better, but yes, a lot of the caves were flooded when the burials took place, because these burials are, in some cases, for example, Sanctuary Blue Hole, the burial is over 100 feet deep and pretty far back in the cave. So I have no idea how they got them in there.
I don't either.
Another question there. What does this mean for modern Bahamians? Can any trace ourselves to the ancient Bahamians?
I can actually answer this one. So I did a study on Y chromosome and autosomal, which is inherited from both the mother and the father. The Y chromosome, there's no remnants of the Lucayans left in the modern population. I did not sample the south, so it might be there, but I did sample the north and central. I did 6 different islands and I didn't find them. In the autosomal, I did find some East Asian and Native American and East Asian influence. So that goes to tell me that it's probably in the mitochondrial DNA. It just hasn't been done for the Bahamas as yet, for the modern population. And that's hopefully a step that we will take after this.
Okay. Any more questions? Anybody wants to directly ask a question? If you don't want to put it in the chat.
So how did you guys know?
How did you guys know about it?
I assume that's a question about the burials in the blue holes.
Yes.
Well, there are a lot of researchers who cave dive, and they came across the burials during cave diving. And to answer the question that will probably follow this, how do we know it's not a modern day burial? The Lucayans practice something called cranial flattening. And so their foreheads are flat, or have been flattened. And so it stands out as a historic burial or Lucayan burial instead of a modern day burial. But there are a lot of people who study the blue holes and so forth. I've been fortunate to go in a few and want to get back in them soon.
Okay. Any more? We are 15 minutes past the time. There's no more questions. I'd like to thank everyone today for this excellent presentation by both the researchers. Thank you once again to our collaborators, international collaborators, for working with UB in this project. Thank you as well to Dr. De Luca for his presentation today. Thank you to all of you for coming for this session today. This session has been recorded. As long as you have registered for this session today, you will get a link to the presentation after this presentation. So give us a few minutes and we will send it all to all of you here. So if you have further questions, please do not hesitate to send the email to me at research@ub.edu.bs, and we can direct the question to the researcher. Or if you are able to communicate directly with the researcher, please feel free to communicate as well. So once again, thank you. We will see you again for our March Research Edge session. Look out for the flyer that will be coming out very soon. So have a good weekend. Stay safe as usual. So bye-bye.